Hello everyone, in this video we're going to be going over the permeability of the lipid bilayer that's seen in both prokaryotic and eukaryotic cells from the notes in my microbiology class. So, the bilayer's permeability is selective, so that means only small molecules such as O2, CO2, N2, as well as hydrophobic actually pass through alongside water. And speaking of water, we do have some proteins called aquaporins that will allow for the passage of water. Other molecules aren't listed here, so ions, large hydrophilic molecules, all of those, they cannot pass through the permeability of the bilayer by themselves. So we need some other methods in order to move them. So the actual layer itself, it's a bilayer where you have the polar heads of the lipids on the outside. The tails are nonpolar and they're in the middle. Sometimes you'll have proteins stuck, and there's a ton of different proteins that you'll find in the bilayer. These small molecules will go through simple diffusion. So regular diffusion is from high to low concentration, and the speed depends on this concentration. Osmosis is the diffusion of water, as seen in many other science courses. We have unequal solute, and we also have the relationship of water. So let's say, again, these are the three states in osmosis. This one where water's moving into this cell that has a lot of solute inside, this is going to be hypotonic. There's more water going inside to where there's less water or high concentration of solute. The situation where they're equal, this is isotonic. And then where water is leaving for the outside that has a higher concentration of solute, this is your hypertonic. When water flows in hypo, the membrane is forced against the cell wall of the bacteria, so the membrane is really pushing against that cell wall. However, in hypertonic situations, that inner membrane, the cytoplasmic membrane in our bacteria, is going to pull away from the cell wall. Energy transformation in the cytoplasmic membrane of prokaryotes happens via the electron transport chain. Sorry about that. I keep fumbling words, sorry. Transport chain. And this is in the membrane, so in the membrane, energy is converted into ATP, and the protons are moved out by electrons. This creates the electrochemical gradient, and the energy harvested is the proton motive force. And this allows for the synthesis of ATP, or energy molecule, and as well as some forms of motility. So most of the molecules that we did not list up here, so ions, large hydrophilic molecules, just large molecules in general, need the help of proteins in order to enter. So the proteins themselves act as selective gates with a high a high quality of specification for a certain molecule or a product. So these transport systems are known as permeases or carriers. And usually you'll have the bilayer, a protein here, in its resting state. And you have our molecule of interest. The molecule will bind to one of the active sites of this protein. And when that happens, we get a conformational change. And then after that, the molecule will be released into the inside of the cell. In these types of transport, we have facilitated, diffusion, which is again a high concentration to a low concentration. However, this time we get the aid of a protein. And this is good for bacteria in low nutrient environments. We also have active transport. This is the one that does require energy, usually in the form of proton motive or ABC transporters and binding proteins. So ATP with binding. The interesting thing about active transport, it requires energy because we are going from low to high. This is a movement against the gradient, as taught in other biology classes and other science courses. Active transport requires energy because you're moving low to high. There's also group translocation. This means the compound is altered. A common example that will be taught in a lot of courses talking about different biosynthetic pathways and whatnot would be phosphorylation. Giving molecules a new light, you're adding a PO group to them. Specifically, there's one of these processes that happens with glucose. We also have things such as protein secretion, in which a protein which is synthesized inside will be phosphorylated by ATP into ADP, so that phosphorylates that protein. It goes through this other channel protein here, a transport protein, and once it has this signal sequence recognized by this protein here, it actually then becomes active and folds into an enzyme once it's outside of the cell. Now, let's say there's a molecule here that is of interest to the cell. That protein as an enzyme is going to work on it, and it's going to chew up the molecule into its monomers, its subunits, and these will then go into the cell via that protein. That's it on the bilayer. Thanks so much for tuning in. Please do something nice for someone.